Well, it's wonderful to be back. I was here with Rachel Dutton back in 2012, which seems so long ago. And back then, I feel like we knew nothing, and we know so much more about this wonderful world of cheese rind microbiology, and that's what I'll be talking to you about today. And the overall goals in my lab, which I now have at Tufts University, are to really understand fermented foods as a whole. So we don't just study cheese, we study sourdough, kombucha, sauerkraut, and a variety of other fermented foods, looking for general principles across these foods. And the big questions we ask are simply what microbes are there, what is the microbial diversity of these foods, which we still don't know for many of these food products. The second thing we want to understand are what are the processes that control the diversity of microbes in those foods? What things are happening in these systems that determine what kind of microbes you get? And then finally, because we're interested in helping food producers, how can we control those processes? How can we use this knowledge to improve both the safety and quality of fermented foods? And a lot of the things that we think about in my lab are actually basic ecology and evolution. I just want to give you a quick overview of how we think about these systems. The first thing we think about, as we were just seeing with the spraying application in these barns, is dispersal of microbes. Microbes getting from the world around us to the particular environment that we're interested in. And of course, in many of these cases, it's cheese. And of course, as you can imagine, it's a really hard thing to see because we can't see microbes, but it's something we think a lot about in the lab. The second thing we think about is what we call selection. And selection can be microbes fighting with each other, so microbe-microbe interactions, both war and peace, as well as microbes interacting with that environment that they're living in. So this could be a piece of cheese, it could be a plant leaf if you're thinking about sauerkraut. So that's the second thing we think about. And then the third thing that we've been thinking a lot about recently, and I think is really exciting, is diversification or evolution. So if you're out making cheese in a cheese cave, you're evolving microbes. Every day as you put new batches of cheese into that cave, you're providing opportunities for microbes to evolve over time. And I'll give you an example of how we're seeing that happening in the cheese environment. So if you put these things together, these are really the building blocks, the basic design principles to how to build a fermented food microbiome. And we're thinking about this, as I said, across many different systems. And fortunately, the food producers of the world are using top-down engineering to make these fermented foods. And what we can do is sort of lift up that veil, a very sort of top-down approach, and think more bottom-up and understand how each of these processes is operating to give us all these beautiful fermented foods that we enjoy every day. I'll only be talking about surface ripened cheeses today because this is the Science of Artisan Cheese conference. Maybe one day there'll be a Science of Artisan Sauerkraut conference and you all can come to that and learn about some of our other work. So I think I don't need to tell all of you that these are cheeses that have a rind growing on the surface, a natural rind, a bloomy rind, or a washed rind. And this is what we presented, even a cruder version of this actually. I was looking at the video this morning with kind of shock and horror of what we learned from our big survey that we did of cheese rinds around both Europe as well as in North America. So these are the data we presented back in 2012. So on the top here, we have data for bacteria, and on the bottom here, we have data for fungi. And each column here represents one of 137 different cheeses that we sampled. And the different colors that you're seeing here represent different genera of those bacteria and fungi. So thinking like oaks, pines, and maples, so at a pretty high level. And so this is what we figured out, and it took a lot of work to get here, and what we've been doing ever since is understanding this diversity, right? So, for example, why do these cheeses over here have a lot of these bacteria that are encoded in red, which represent various types of proteobacteria? Or why do these particular cheeses right here have a lot of Staphylococcus species? And these are the puzzles that we've been sort of going into in the lab and trying to pull apart over the last 6 years. So I'm going to present to you 4 quick lessons from the past 6 years about what we've learned and where we're going. And the first thing is that microbial interactions are really diverse and are important drivers of cheese rind development. And I think many of you know this, right? You select particular starter cultures to get a particular rind, but what we've been figuring out is why do you actually select those starter cultures and what are they doing? The second thing I want to point out is that cheese microbes can rapidly evolve. Over a period of weeks, your favorite mold in your cheese cave can become a totally different mold with completely different traits, and I'll show you an example of that. The third thing is that we need to appreciate that strain level variation. So within a species of a microbe, the variation in traits that can exist, that's really important for how our cheese communities are developing. It's something that we didn't really think about 6 years ago, and we were looking at that high level, but we're really appreciating today. And finally, we're getting into quality, thinking about how things can go awry. As you all probably know, there are many things that can happen along the way, and how can we address some of those problems with the knowledge that we've been obtaining? So here we go. So microbial interactions. We've been thinking a lot about bacterial-fungal interactions. So there are both molds and yeasts, so those are the fungi of our cheese rinds, that are interacting with a whole suite of bacteria in those rinds. And so what my lab has been doing is sort of going in and looking at various examples of these interactions. One really cool one that we found is that bacteria can use molds as superhighways. So bacteria in really wet cheeses, especially washed rind cheeses, and even some natural rind cheeses, they can actually jump on the hyphae, the filaments of those fungi, and cruise around in them like a superhighway. And that can dramatically change the type of bacteria that you'll get on your particular cheese. And I won't talk about that today, but you can check out more of that work, and I can talk more later if you'd like. Another thing we're finding is that there are a lot of antibiotics being produced by fungi on cheese rinds. So we heard about penicillin earlier. There are actually penicillin-producing fungi growing on many cheeses that we sample. What you can see here, this is a fungus that's killing off that bacteria, and there's a little zone of inhibition that we're seeing here. And so there are a lot of these examples of bacteria being killed by neighboring fungi. I'll quickly talk about an example today about how volatiles, the things that we smell, are ways that microbes are communicating with each other, and that can again impact the type of rind that you might get on your particular cheese. And a couple other things we're learning about are things we already sort of knew, that proteolysis, the breakdown of proteins, can facilitate microbial interactions. And more recently, we've been finding that bacteria and fungi are fighting for metals. So things like iron and zinc are really important in those early stages of rind development. So let me just quickly, this is a whole snapshot of all the different things we're thinking about. Let me just give you an example from this really cool recent work on volatiles. This is work by a graduate student in my lab, Casey Cassetta. And what we were thinking about in the world of microbe interactions, people are talking about how volatiles, the chemicals that are produced by microbes that are in the air, can sometimes be signaling molecules. And we thought, well, cheese stinks, right? Cheese is a very potent, very volatile environment. What could be going on in cheese? So what Casey did is she set up these really cool experiments where she had microbes, various bacteria, that are responding to the presence of volatiles produced by fungi. And when she does these experiments, we make a cheese sandwich in the lab where we put these microbes next to each other. They're not touching each other, but they can smell each other, right? They're right next to each other, and they can respond to the volatiles being produced by their neighbors. And so Casey was specifically interested in how bacteria respond to fungal volatiles. And so what I'm showing you here are the data for all these different responders in the presence of these various fungi. And what you need to know is that red means growth promotion. So these are bacteria that are strongly upregulated. They're very happy in the presence of the volatiles, these aroma compounds produced by the fungi. And one thing I'll point out, this bacterium called Vibrio, Vibrio casei, which is really widespread in washed rind cheeses, loves the volatiles produced by fungi. So you'll see this is bright red for all these various fungi that we're working with. So that means when Vibrio is growing next to, but not touching, fungi, if it can smell those fungi, it grows a lot more in the presence of those volatiles. Super cool stuff, right? Why is it doing it? We're trying to figure that out right now. And this is, I'll point out, this is pairwise interactions, right? This is one fungus and one bacterium. What happens if you try to mimic a whole cheese rind community, right? As you do in your cheese caves every day. Well, we did that experiment. So we actually mixed all the bacteria together instead of doing it in individual combinations, and looked at how maybe Vibrio might take over in these communities in the presence of fungal volatiles. So again, it's the same experiment, these little sandwiches, but with a mix of bacteria. And this is what the data look like. So this is the different treatments. This is a control community of bacteria without any fungal aromas, no fungal volatiles. And then these are the various experimental replicates that we have in these different treatments with the fungi. And what you'll see is Vibrio doesn't do very well. This bacterium that responded strongly in the experiment before, it doesn't do really well without fungal volatiles. But as soon as you add Deberomyces or Geotrichum volatiles into these experiments, oops, sorry, I'm shooting the laser at you guys, that's bad. It takes off in these communities. So I think this is really cool because when I walk in a cheese cave, I start to smell all these aromas and these volatiles being produced. And this could be evidence for sort of a positive feedback, right? If you start growing a lot of geo cheeses and you put in a new batch of cheese that has just a little bit of Vibrio there, it might take over versus if it didn't have any of these volatiles present. So I think there are some interesting lessons we can learn here in the context of managing these cheese aging environments. So that's just one example of how these interactions between microbes, and not even direct interactions, but through the air, can affect these rind communities. So let's quickly think about how cheese rind microbes can rapidly evolve in these environments. So microbiologists have been thinking about bacterial evolution in part because of antibiotic resistance. We know that bacteria can quickly evolve the ability to fight back and not be susceptible to these antibiotics. And when I think of a cheese cave, I think of these same kinds of experiments where you're continuously putting in new batches of fresh cheese that bacteria and fungi can colonize, grow, and then recolonize and grow. This is like evolution that's happening inside these cheese caves. And so you can start off with these natural system environments and in these wild populations of microbes, and over time they could evolve to live in cheese. And this is an idea that's been out there. People have thought about this with beer yeast, and they've thought about it with koji, the thing that we use to make sake, but it hasn't really been demonstrated with cheese microbes. And we've been specifically interested in penicillium because, as we know, the beautiful white wheels of Camembert and other cheeses where we use Penicillium camemberti, historically they would have looked more like this, right? So back before we had this beautiful mutant starter culture, this is a mutant, by the way. You can't find this in the wild. The cheeses that you would have bought in France 200, 300 years ago would have been more like this over here. So they would have been bluish green, they would have been really musty, sort of smell like a basement. And as we learned about, there are mycotoxins in the world, and probably 200, 300 years ago, Camembert was a little bit toxic. And of course, today we don't have any of these toxins. So how did we get from here to here? There's some work, there's some notes sort of stuck in history about how Pasteur's students brought us these particular strains that we use, but we don't know the process of how they evolved. And what's really interesting to me is that if you sequence the genomes of these sort of wild penicillium strains, so the things that grow on our natural rind cheeses or those really annoying little blue spots that grow on our bloomy rind cheeses, they're super closely related to the thing that we use to make Camembert. So they're really close relatives. So you might hypothesize that this is just simply a domesticated version of this. And that's something that we've been looking at in the lab. So what we do is we take wild penicillium molds, these blue molds that are out in the cheese caves, and we just grow them over and over and over again on cheese. And we just let them sort of grow and evolve and see what they do. And this is work that I had an undergrad in my lab do. So this is Ina Bodonaku who started this work. So we started off with a wild ancestor penicillium that looked like this. And what really struck me is that over just a period of 4 weeks, we ended up evolving these mutants, these things that look a lot like the thing that we now use in Camembert. So this is just showing you the data from that experiment. So this is over, again, just 8 weeks. Over that period, we get about 80% of the populations are being taken over by these white mutant molds, which is really cool. This is happening over just a period of 8 weeks. If we look at these mutants and sort of look at all the traits that they have, we notice 2 really striking things. One is that the mutants don't make toxins anymore, so they completely shut down mycotoxin production, which is a good thing, right? We don't want toxins in our cheese. And that means they're just sort of letting go of this really expensive process of making toxins. The other thing you can see, of course, is that they're no longer pigmented, and they also stop making a lot of spores. And these are all traits that are costly. Making spores and making toxins takes a lot of work. So if you're in this delicious, stable cheese environment and you're constantly being exposed to that, you let go of these costly traits and you focus on doing much more simpler things and living a simpler life. The other thing that Ina and I noticed when we were working with these strains is they smelled amazing. So that ancestor we started with smelled like my basement, really musty, really nasty. And then the evolved strain smelled like good cheese. We were working with the petri dishes, and we kept getting wafts of blue cheese and camembert. So we worked with a chemist to actually measure this, to actually understand what are the chemicals that these different fungi are producing. And so this is a whole suite of compounds that these fungi can produce. And what I'm showing you, this is the wild type right here, and then these are 3 of the mutants. The yellow color means it's making more of that particular compound, and blue means it's making very little of that compound. And what I'll point out is that the ancestor makes a lot of this compound geosmin. Geosmin is that smell of earth after it rains that a lot of microbes can make. So the ancestor makes a lot of that, and then these evolved mutants make none of that. So that's where the mustiness is going away. And then the cool thing is that these evolved mutants, the things that have been domesticated, make a lot of these ketones. We know ketones are the flavors of blue cheese and a lot of other great aromas from cheeses. So again, domestication have shifted these molds over just a period of weeks from going from this musty basement critter to something that is very similar to the thing that we now use to make cheese. This is not just a lab phenomenon. So one critique of experimental evolution, which microbiologists do all the time, is, well, that's great. It happened in a petri dish, but does it happen in nature? And when we did a deep dive up at Jasper Hill and we sampled their molds that we know very well, we do find at a very low frequency some of these molds popping up, these white mutants that are identical if you sequence the whole genome to this, but they have these very different traits. So they're out there. They're in your cheese caves. You're evolving these things over time, and they might be really useful for cheese making in the future. That was the story from Penicillium. We're actually also looking at how Staphylococcus species evolve. And here it gets a little bit more interesting because there are some traits in staph that we do not want to evolve, like antibiotic resistance and making biofilms. And we do see that happening in cheeses as well. So I won't tell you that story, but I'm happy to talk about that later. Okay, so microbial interactions are really important, and volatiles can mediate them, and microbes can rapidly evolve. What I want to do is just quickly end with the last 2 points about strain level variation and how microbial shifts can impact cheese quality. So the last time we were here, Rachel and I told you that you can make a cheese in California, Vermont, France, and Wisconsin, and they can all have about the same microbial profile. So we sort of shot down this idea of microbial terroir. And there was a lot of sadness, and I understand that, because you all want to have unique microbes that are a sense of your place. I'll tell you, we've revived microbial terroir. So keep in mind that this is at the taxonomic level of genus, right? So we were just thinking about maples, oaks, and trees, Brevibacterium and Geotrichum. We weren't thinking about the species or strains. So what we've done is we've gone in and tried to experimentally test if strain level variation really matters in cheese making. And the way we've done this is we went out to 9 different cheeses that we had sampled in our survey, everywhere from in Oregon to Spain. And in those 9 cheeses, we isolated the same 3 bacteria, the same exact species, right? So from each cheese, we got Staphylococcus aureum, Brevibacterium, and a Brachybacterium. And these are really widespread in cheese making. You all probably have them if you're making a natural rind or a washed rind cheese. Okay, so from each of these 9 cheeses, we have these same 3 species that have probably evolved in each of those environments. And what we can do is we can sequence the genomes, the entire genomes of those strains, and we see huge variation. They're the same species, right? They're the same name of bacteria, but in terms of what they can do, it's wildly variable. So in terms of how they use carbohydrates, for example, across these 9 communities is very, very, very different. So their genomes are totally different. But the other thing we can do is we can take those strains and put them in a common garden environment in the lab. So we could take all those strains and give them our little cheese curd medium that we use and ask, does each cheese behave similar, right? So do those strains from those different environments do the same thing, or do they behave very differently? So they're taxonomically the same, but are they functionally different? And so what we can do then is look at how the community builds over time. So this is looking at how we initially set up the community with each microbe present in the same amount. And we can look at how they evolve. These are 5 different replicates to show you that it's robust across individual cheeses. And then this is over time. So this is about day 10. I realize that 10 days of a lab cheese is not equivalent to 21 days or 2 months of a field cheese, but this is just for proof of principle. So that's for one cheese. This is what it looks like across the 9 different communities. So what I want you to do is look down here at day 10. You'll see some cheeses are very dominated by Brachybacterium. So this community 4, for example, from one part of the world, ends up having lots of Brachybacterium. Others have a lot of Brevibacterium, and some sort of have coexistence of all 3 strains. The other thing I'm showing you down here, this is how the cheese looked. This is just looking top down at it. So, for example, this cheese is super pigmented, really, really orange, whereas some of these other rinds were more sort of reddish or sort of beigey colors. So if you have exactly the same species of microbes in different cheese-making parts of the world, you can end up with totally different cheeses. So hooray, microbial terroir is back, yay! The other thing I'll tell you is when we smell these, and we're working on, again, the volatile assays, and when we smell these in the lab, they smell totally different as well. And I know many of you use different strains, and you think about strains. So what we're trying to point out is that the strain level variation matters. Okay, and finally, a quick quality story. So we get Franken cheeses sent to the lab all the time now. We get these cheeses that have gone off in non-desirable directions, and we love this. This is super fun for us because we can use that baseline information that we've collected from what is a good cheese and then figure out what's there that shouldn't be there, or what's changed. And we'll talk later on today about how you can set up experiments to address that. My favorite was this story right here. So this is what we ended up calling purple rind defect. Rinds shouldn't be purple. We had never seen this before. And this was a cheesemaker in the US that was using raw milk. And we first got this cheese sent to us in 2015. I called up everyone that I knew that would have ever seen purple rind defect, and no one had seen it before. So we got really excited about characterizing this. So this cheese normally looked like this, and was looking consistently across many batches like this. And entire wheels would be purple. It wasn't just patches. Every single wheel of this cheese turned purple. We played it out in the lab, and again, we can use the baseline knowledge we had of what was there and what wasn't there. And these are just colonies on standard plate count agar in the lab. And this is the typical cheese. You got your Brevibacterium here. These are Psychrobacter colonies. There's your Staphycorum. And what was really striking to us is every sort of patch that had lots of purple had these wavy patterns going out, and that's the bacterium Proteus. And Proteus has been found by the French on lots of different cheeses in relatively small amounts. It makes really strong, sort of like fishy sulfur compounds. No one ever really described it as being bad or causing problems for cheese, but, sorry for the gross picture here, but in the literature, people have described this thing called purple urine bag syndrome, or PUBS. So this is where people are in a nursing home or in a hospital, they have a urine bag, and suddenly, and very shockingly, it turns purple. And that's because they have a urinary tract infection with bacteria that can make indigo. Indigo and a close compound, indirubin. Bacteria can make the compounds we use to dye blue jeans. And when you mix blue and this sort of reddish color together, you get purple. And so what we're finding is, and we've proven now, that this bacterium that was growing on this cheese, this particular strain of Proteus, can make indigo, and that's what it was doing on this particular cheese. It was using the tryptophan, which is an amino acid in casein, to make this indigo compound. We were able to then recreate it in the lab. We followed Koch's postulates, and we were able to demonstrate that when we added Proteus to our little lab cheeses, it turned purple. And we did a bunch of chemistry to demonstrate that it, in fact, was indigo. And this is a paper that'll be coming out in Food Microbiology in a little bit. So we can use that background data to solve cheese problems. Now, this was a really dramatic one. Like, purple is a very striking cheese defect. But we've done this with some flavor defects, and we can do this with a lot of other variety of quality issues that folks are having. So microbial interactions are out there, and they're playing really important roles, even through volatiles, through the air that you're breathing in your cheese caves. Microbes are rapidly evolving, and we're looking at lots of different examples of that. Microbial terroir is back, and these shifts in microbiomes can impact cheese quality. So with that, I'll thank all the wonderful people in the Wolfe Pack that helped make all this work come together. I'll thank the USDA and the National Science Foundation in the US for funding this really cool food work, but for the basic science. And then, of course, Bronwyn and this whole event for inviting me to come back and tell you what we've been up to. Thank you.